Welcome to this explainer. Today we're diving into the absolute magic of fermentation. We hear that word tossed around all the time, right? Usually it's about sourdough bread, craft beer, or maybe kombucha. But what we're looking at today goes so much deeper than your kitchen counter. We're going to explore how science took these everyday microscopic bugs and turned them into massive industrial powerhouses that literally shape our modern world. So, let's start with a really big question. How exactly do tiny, totally invisible organisms end up manufacturing our antibiotics, our sustainable biofuels, and yeah, even the bread on our tables? I mean, it sounds kind of like magic, doesn't it? The idea that something 1,000 times smaller than a grain of sand is out there synthesizing complex, life-saving medications, but actually it's not magic at all. It's highly controlled, incredibly precise biological engineering. To get there, we'll be following a pretty clear roadmap today. First, we'll look at the magic of fermentation itself. Then we'll take a brief look at the scientific history. Third, we'll break down the fermentation formula. Fourth, we'll see how we scale up the whole process. And finally, we'll wrap up with why all of this actually matters to you and me. All right, let's jump right into section 1, the magic of fermentation, and specifically the difference between the biological and industrial sides of it. You see, fermentation actually has a bit of a split personality, depending on who you ask. If you talk to a biologist, they see fermentation strictly as a metabolic process. It's just a way for cells to make energy, or ATP, without using oxygen. Pretty straightforward. But if you talk to an industrial engineer, they see it totally differently. To them, fermentation is a large-scale, highly controlled biological factory designed to pump out valuable goods. The wild thing is, it's the exact same underlying mechanism. We're just looking at it through two very different lenses. One is all about cellular survival, and the other is all about human utility. To really picture that biological view, imagine a microscopic yeast cell just doing what it does best, eating. It scarfs down glucose to create that ATP energy for itself. But the beautiful part for us humans, it's the waste it leaves behind. As it processes that sugar, it spits out ethanol and carbon dioxide as byproducts. That basic, microscopic act of eating and excreting is literally the foundational engine of the entire biotechnology industry. But here is where we step in and start calling the shots. When we harness these microbes, we can manipulate their environment to get completely different products out of them. For instance, we can run an aerobic process. That means we constantly pump in oxygen. This gives the microbes a huge energy boost, making them multiply like crazy, which is exactly what we want when we're trying to produce things like the antibiotic penicillin or industrial enzymes. On the flip side, we can go with an anaerobic environment. By completely cutting off the oxygen, we force the microbes to use different chemical pathways just to survive. And that specific survival mode is what allows us to brew alcohol, create lactic acid, and generate sustainable biofuels. Moving on to section 2, let's take a quick look at the scientific history and how this went from an ancient art to a precise science. For the longest time, fermentation was basically just an empirical art. People knew that grape juice turned into wine if you left it alone, but they had no idea why. The massive turning point hit in 1857. That's when Louis Pasteur came along and proved that living, breathing microbes were actually causing the fermentation. He completely shattered the old idea of spontaneous generation, famously describing the process as life without air. And then, a few decades later, Edward Buchner figured out something even crazier. You didn't even need the living cell. The enzymes inside the yeast could actually do the job all on their own. When you actually trace this timeline, it is truly wild. We start way back around 6000 BCE with ancient Mesopotamian brewers just crossing their fingers and hoping for the best. Fast forward to the 19th century, and we've got Pasteur's microbial theory. Then, boom, 1928, Alexander Fleming discovers penicillin. That discovery triggered the creation of massive, industrial-scale submerged fermentation factories in the 1940s, literally saving countless lives during World War II. And finally, that brings us to the late 20th century with the absolute game changer, recombinant DNA. Suddenly, we could genetically engineer microbes to produce things they would never make naturally, like human insulin. Okay, let's dive into Section 3, the fermentation formula and the actual engineering behind the process. I have to say, I absolutely love this equation. It elegantly boils down a wildly complex industrial system into 6 straightforward variables. If you ever wanted to build your own biomanufacturing plant, well, here is your blueprint. Let's break down exactly what each of these 6 pieces actually does. First, you take your chosen microorganism, maybe a bacteria, a yeast, or a fungus, which is going to act as your biological catalyst. Second, you feed it a substrate. This is just raw material like sugar, starch, or even agricultural waste. Third, you put all of that into a bioreactor, which is the vessel where the magic happens. Fourth, you tightly control the conditions inside that reactor, things like temperature, pH, and oxygen levels. Fifth, you introduce an inoculum, which is really just a healthy starter culture to get the whole process moving quickly. And finally, step 6 is product recovery. That's where you extract and purify your valuable product at the end, separating it away from all the microbial waste. Now, for Section 4, we're looking at scaling up the process. How do we go from a tiny lab flask to a giant factory? When you scale this up, that simple little glass flask gets replaced by a highly engineered, absolute behemoth of a bioreactor. Because when you jump up to thousands of liters, those trillions of microbes generate a massive amount of heat, and they need a perfectly uniform environment to survive. So engineers use these giant stainless steel vessels rigged with heating and cooling jackets just to regulate the temperature. They've got computer probes constantly monitoring the pH and dissolved oxygen in real time, and giant paddle stirrers keeping the whole mixture perfectly agitated. That way, every single microscopic bug gets exactly the food and air it needs. Now, depending on what exactly we're trying to make, we run these giant tanks in 3 completely different ways: batch, fed batch, or continuous cultures. And the volume of liquid inside the tank actually behaves very differently depending on which method you choose. Let's break those down. Batch fermentation is basically a closed system. Think of it like making a single loaf of bread. You throw all your ingredients in at the start, let it run until the food is gone, and then you harvest. It's super simple and has a very low risk of contamination. Then you have fed batch, which is semi-closed. Here we carefully add nutrients over time. This keeps the microbes in a highly productive state for a lot longer, which really boosts yields for complex products like penicillin. And finally, continuous fermentation. This one literally never stops running. You are constantly pumping in fresh food and constantly siphoning off the final product. It gives you maximum output for bulk things like industrial ethanol, but because the system is open indefinitely, it carries a much, much higher risk of contamination. Now, if you do decide to run that continuous system, you have to control it carefully so the microbes don't overpopulate and totally crash their environment. You can do this in one of 2 ways. You can use a chemostat, which limits a very specific nutrient. That effectively puts the microbes on a strict diet to control how fast they grow. Or you can use what's called a turbidostat. This uses sensors to constantly monitor how dense or turbid the liquid is getting. If it starts getting too cloudy with microbes, the system automatically adjusts the feed and dilutes the mixture to thin the herd. It's a brilliant way to maintain a constant maximum growth rate. All right, that brings us to our final section, why fermentation actually matters, and its massive impact on our world. So why should we care about all this microscopic plumbing? Well, it matters because this process is literally the backbone of our modern world. I mean, sure, it gives us the bread, cheese, and soy sauce in our kitchens, but it's also the biomanufacturing base for our entire healthcare system. It provides the penicillin to fight off infections and the recombinant insulin that millions of diabetics rely on every single day. On top of that, it's producing sustainable biofuels and industrial chemicals that are actively helping us reduce our reliance on toxic petrochemicals. Even environmental engineers are using large-scale anaerobic digestion to treat our waste and turn it into usable biogas. It is everywhere. And honestly, we're just getting started. As we begin to engineer these microbes with advanced genetic tools like CRISPR, we're stepping into the era of synthetic biology. Right now, we're using precision fermentation to create real dairy proteins without ever needing a cow, and we're engineering organisms to spin entirely new, sustainable synthetic materials. So understanding the incredible power of the bioreactor leaves us with one incredibly thrilling question to ponder. As we get better and better at this, what world-changing product will these tiny, invisible factories build for us next? Thank you so much for joining me for this explainer. I really hope it completely changed the